Hello everybody, Sunday again. Okay, we are on A2.2 cell structure, SL and HL. Very long section. Hope I can finish it once. Okay, okay, let's go. And we are on the A2. Okay, unity and diversity in cells. Okay, this section talk about the cell structures. Okay, first of all, we have the cell theory. What's the cell theory? Cell theory basically is the point 5. Okay, cells only come from pre-existing cells. Okay? It is a theory to explain how cell can evolve from cell and why can't the cell evolve from non-cell. Okay, this is the concept. Okay, and Pasteur's experiments disprove the spontaneous generation of life. Okay, so we have a new theory. So the cell theory is the next one for spontaneous generation of life. Okay, and by the cell theory, we predict all other new cell appears is come from the old cell. Okay, so it is a theory. It is a quite good theory because it can explain most of the observation and can have some prediction. And when we want to study the cell structure, we need to use a microscope. In using a microscope, we need to concentrate on the scale bar. Okay, and then of course you have done it already. Okay, in IBDP you need to do a lot of experiments on cell. Okay, first of all you need to mount the cells and tissues. Okay, you need to stain it. Okay, and put it on the table. Okay, and then you need to focus. Okay, by the coarse and the fine adjustment. And then after that you find your specimen, you find the cell. Okay, and then you need to calculate the actual size and the magnification. And for some microscope nowadays very powerful, can take some photos. For example, it is a typical cell, a typical thing in a micrograph. Okay, you can see a cell, you can see a scale bar, and you can measure it. Okay, after taking a photograph, okay, you measure the length of the cell by the ruler. Okay, find the length. Okay, and then you find out the magnification. In IBDP, it always represent the magnification by x 200, means 200 times the of magnification. And first of all, you need this magnification about the linear scale magnification, about the length of the image over length of the real specimen. If magnification is larger than 1, it means that it is a magnified graph. If magnification is smaller than 1, this is actually restrained, diminished. And when we're using a microscope, okay, of course magnification is larger than 1. When you are drawing a microscopic daily life object or someone taking a photo, okay, in daily life, the magnification is always smaller than 1. Okay. Remember, same ratio of length for a photograph. Okay, so when you talk about the length ratio is the magnification. You talk about the LVL ratio will be the magnification square. Okay, in IBDP, remember, you need to do a few steps, okay? First of all, you need to measure the length, okay, of the photo zones, okay? And then you have to divide it by the magnification to find out the real length of the specimen. And of course, sometimes you need to find out the magnification too, okay? You look at the ruler, you use the ruler to find out the real length of the scale bar, and then the scale bar has a length also, okay? And then divide the two things like this, again, you'll find the magnification. So to that, find the magnification. Next step, find the real length of the specimen. And the error is fairly teeny tiny, just 0.1 cm measurement. For when you use the ruler, make sure you measure it accurately. And for a modern microscope, wow, very strong. I learned a lot of modern microscope and skill. Okay? For instance, you need to learn that electron microscope. You use the electron to reflect the thing from the specimen, and then to find out what happened to the electron to deduce what you can observe. The resolution, the magnification can up to 10 million times. Okay, but the disadvantage of electron microscope is very expensive. You don't have it in a school lab. Okay, you need to have it in a university level, and very costly. Absolutely not portable. A big machine actually. And then you need a lot of training to use it. And it is not for living specimen because if the specimen move, you can't focus it. Absolutely, because you're looking at a teeny tiny place, okay? Because it is a 10 million magnified, okay? And we have scale called freeze fracture. Okay, so we want to look at the three-dimensional structure of a sample, but actually we just have a surface, right? So we freeze the sample, we break it down into different planes, okay, and then we observe different planes of the sample, and then join all the planes image together, and we can find out the three-dimensional photos or three-dimensional structure of the sample. So we call it that freeze fracture method. It is used for electron microscope. First of all, we freeze the sample and then break it into different parts, okay, and then they have different plane of the samples. Okay, we look at each plane, each fracture planes one by one. Okay, by the electron microscope, that's key structure for the cross section. Okay, in biological application, we call it cross section. Okay, so join all the cross section together, join all the wheel of the plane wheel together, and then you find the internal organization of the membrane of the thing. Okay, means the internal 3D image. Okay, free fracture. Remember, break in the planes, observe the plane wheel, join together from the 3D structure. Next one, synogenic electron microscope. This thing is to freeze the thing into a very low temperature, okay, negative 170 degrees Celsius. Okay, so make sure everything is not moving. Okay, so you can see the structure very clearly. So remember this one, synogenic, remember, very low temperature. Fluorescent, you know fluorescent, right? Give you some stain, okay, and then under the light microscope reflection, and you can see the highlighted parts, okay. So we have different brand of the dyes, okay. This is Alicelfra is popular one. You don't need to remember the name, okay. So so just stain the specific parts of the sample, okay, and then you can see it clearly. For example, for the bacteria cell, okay, you put the thing inside, and then the thing will the dye will go to the bacteria cell, and it will be highlighted, and you can count the number easily, more accurately. The other one is immunofluorescence in the light microscope. How to find out the cell's immune system? You remember later on the cell there is something called antigen. This antigen show that the cell is foreign to the body, and this antigen will be caught by the antibodies. And if you have a dye that can cause these antibodies, so the dye stick to the antibody and the antibody stick to the antigens. So the antigen will be highlighted, and you can see clearly the antigen on the cell. Okay, this is the procedures. Okay, so it is a lot of technique. Okay, we don't do it in school lab. Okay, because immunology. Okay, about bacteria, about virus. Okay, we don't do it now. Okay. Okay, common structure of cells. Okay, we go to the details. Okay, there's a common structure for every kind of cell. Prokaryotic, eukaryotic, okay, single cell, multiple cells. Okay, all cell has a plasma membrane. Remember, you have to separate inner inner environment from the outside environment, right? The cell membrane separates the cell from the surroundings. Okay, so inside the cell membrane there's a cytoplasm. Okay, it's mainly of water, and the cytoplasm is the space. Okay, the fluid inside the cytosol. Okay, inside the cytoplasm there is a DNA. Every cell has DNA. Okay, there are different ways to store the DNA, and the DNA is a genetic material for heredity. And we have two different kinds of cells. One is prokaryotic. We'll explain it. Okay, what is a prokaryotic? Prokaryotic is a simple cell. Okay? Inside the prokaryotic, there is no compartments. Okay, just one cell inside, no compartment, no further compartmentalization inside. Okay, it means a no organelle inside. And prokaryotic does not have an organelle. Okay, bounded by membrane. Okay, if there is organelle, we call it the eukaryotic. We'll learn it later. And prokaryotic has a cell components, so for example, cell wall, plasma membrane, cytoplasm, liquid DNA, okay, and the 70S ribosome. Ribosome is used to produce protein. Prokaryotic cell structure varies actually, and prokaryotic cell has a smaller size, 1 to 10 micrometer, okay, and it is a unicellular organism. Okay? An example of prokaryotic bacteria, E. coli, okay. And then there is our syllabus for IBDP now. Okay, if you look at the old past paper, they have displayed that this is cut. Okay, for example, the details of the variation of prokaryotic cells. Okay, and this is a typical prokaryotic. You can see a lot of different parts. Okay, we will explain it bit by bit. Okay, for some prokaryote, it can have a tiny and a capsule outside, even outside the cell wall. Okay, this is one is for the increase of the cohesion when it's sticky. Okay, stack up. Okay, or the floors. Okay, cohesion between the prokaryote and the outer surface. Okay, and then it has a flagella. Flagella is a longer one. Okay, short one is a really tiny, and the long one is flagella. Flagella is the movement. Okay? And there's a nucleus. You remember it's a leg DNA. Okay, so it's a single long and circular DNA without protein. Okay, it's a leg DNA. Okay, no nucleus. Okay, nucleus, and then it is a long one, and this is a short DNA. We call it plasmid. Plasmid is a short circular, small circular DNA, and it has a cell wall. Cell wall protects and maintain the shape of the cell. Okay, and there's a plasma membrane. A plasma membrane controls the transfer of material into and out of the cell. And the cytoplasm is the area inside the cell membrane. Okay, and it is filled with cytosol. Cytosol is mainly water. Okay, and it has a 70S ribosome, and it is a size for protein synthesis. Later on, we will learn 80S ribosome. It's just big and smaller. 70S is smaller. You can remember in this way, it's smaller. Prokaryote is smaller. Ribosome 70S. Okay. And naked DNA is a DNA that is not associated with protein. Okay, and the capsule, the tiny nucleus, and plasmid only appear in the prokaryote. So when you saw this four things, confident it is a prokaryote. And the other is a eukaryote. Eukaryote by definition is a cell with compartments. Okay, so you can call it an evolution from a prokaryote to eukaryote if it has engulfed some prokaryote inside and become eukaryote. But this is area we'll talk about later. Okay, the compartments of eukaryote form the organelles in the cell. Okay. It is an inner structure inside inner structure. Okay, different. Okay, so eukaryote has a larger size. Okay, 10 to 100 micrometer, and unicellular or multicellular organism. Remember, so if you saw there is a unicellular organism, it can be eukaryote or prokaryote. Okay, but if it is a multicellular, it must be a eukaryote. So make the difference. Okay, for example, paramecium, animal cell, and plant cell. Paramecium is an example of unicellular eukaryote organism. It has an organelle inside. So the difference between them, okay, you need to bear in mind. Okay, long question, multiple choice, always talk about it. Okay, smaller size prokaryote, larger size eukaryote. Without compartment, this is the definition. Without organelle, with compartment, with organelle. DNA without proteins, the liquid one, the circular one, long circular one, and short circular one. Remember the plasmid, okay, and nucleus. And DNA with protein, okay, we call eukaryote. And circular chromosome like a linear chromosome for eukaryote. Later on we talk about it. Okay, how to pack them. We've talked about how to pack them. 70S ribosome and the 80S ribosome, and then they're reproduced by fission and mitosis. We talk about it in the next lesson maybe. Common in both prokaryote and eukaryote, plasma membrane, cytoplasm, DNA, and ribosome. We have talked about it before. Okay, and only appear in prokaryote and eukaryote. Okay, prokaryote only capsule and remember what did they do? Increase the cohesion with the surrounding. Plasmid, remember it? Small circular DNA. Eukaryote, mitochondria, other organelles, okay, are not. And prokaryote, always remember, without compartment. Eukaryote with compartment. Without is a strong word, must have. With is can have, and mostly have. But if they don't have it, still okay. Can have exception, okay. For example, one prokaryote has low cell wall or capsule. Be careful about this with and without. Without is absolutely low. With is can be or not. Cell structure of a eukaryote, okay. Common feature of eukaryotic cell, okay. Eukaryote, okay. I have a plasma membrane, okay, enclosing some compartmentalized cytoplasm, and it is using an ATS ribosome, okay. And there's a nucleus with chromosome made of DNA, okay. And DNA is bounded by histone. Remember the H core and the histone around it, okay. Remember? And double membrane is very important. Double and membrane and pores. We have a theory on it, okay. And membrane bound cytoplasmic organelle, okay. Mitochondria, endoplasmic reticulum, Golgi apparatus, and the reticular vesicle vacuole, lysosome, okay. Wow, lots of things. You will learn it. You have to learn it. And the cytoskeleton of a microtubule and a micrograph for the structure, okay? Wow, this is the structure of a eukaryotic, and their function is just half of it. Sorry, we're still again. Nucleus is the DNA, okay, with the protein in linear chromosome surrounding histone protein with protein, okay, surrounded packet. We call it not like DNA, okay, it's packet nucleus. And nucleus is the site of ribosome, is the nucleus of the nucleus, okay, inside, okay. And plasma membrane, okay, the same thing, control the transfer of material into and out of the cell, okay. Cytoplasm, okay, the space inside, remember, and the liquid inside is cytosol. Vacuole is the water, okay, store and maintain a water contents. Vacuole is smaller for animal cell, for plant cell is bigger usually. Vesicle is anything store and transport other material, okay. And ATS ribosome size for photosynthesis is bigger, okay. Besides this, we can have other things, okay. Okay, rough endoplasmic reticulum, RER, is the synthesis and modify and transport of protein. Protein is synthesized in the nucleus, okay, and then transport out, and then as lots of further process matured. Okay. And smooth endoplasmic reticulum, okay, synthesized lipid, okay. For the smooth one, with the ribosome, it is synthesized lipid. With ribosome, the rough one is to make the protein mature, okay, or synthesize protein, you can say. Golgi apparatus modify and store protein, okay, and then they will profit over vesicle to transfer protein, okay. Central, okay, central is the only thing that it's only without cell membrane is central. Central, they tell the cell, okay, where is the position of the nucleus and the organelle, okay. And the lysosome transport and store the enzyme, okay. So lots of things inside, okay. Lysosome, enzyme, vacuole, water, vesicle, other thing. In the cell structure of a plant cell is also eukaryotic, but it's special for a plant cell. What's the difference? Look at here, compared with the previous one. A big vacuole, cell wall to maintain the shape, okay, and chloroplast, okay. How to distinguish chloroplast and mitochondria? Can you see? Short line is mitochondria, long line is a chloroplast, okay. Stroma is here, the big dark spot inside, okay. So we'll talk about it later, okay. And then the cell will protect the cell and keep it straight, okay. Starch granule, okay, because it can produce chloroplast produce glucose, and the glucose is stored in starch or starch granule. And then chloroplast, okay, chloroplast carry out photosynthesis only in the leaf cell. I know. And the central vacuole is a big vacuole. There's small vacuole also too. And remember, the vacuole surrounding it is not the vacuole surrounding is the tonoplast. It's a special membrane called a tonoplast, okay. And number of membrane, be careful. This is very important. We have a special theory for that. Okay, low membrane organelle is essential. Remember what they do? Yeah, tell the cell where is the position of the nucleus and other organelle. And single membrane organelle are not. How do you remember it? Just remember the double one. Double membrane organelle just three: nucleus, chloroplast, and mitochondria. They come from other places. Enjoying the outer structure of a cell, we can use single line for single membrane, okay? And close parallel double lines for double membrane. This is the rules, remember, for the perfect one. Okay, 7 functions of life, okay, then we talk about what is life, okay. Life have a 7 function, okay. Nutrient, get nutrition, metabolism, okay, chemical reaction, growth, more cell, size and mass, response, some stimuli, homeostatic, very important term in IBDP biology. Remember, homeostatic, maintain the inner environment situation, okay, environment. Reproduction, okay, reproduce, okay, the cell and reproduce of the organism, okay, as a whole. Excretion, taking care of the something that the waste, so they don't like it, excrete, release it, okay, and harmful or toxic maybe, not useful. And a one cell organism, wow, very hard working because they have to do all the thing in one cell, all the 7 functions in one cell. Remember what the 7 functions: nutrient, metabolism, growth, response, excretion, homeostatic, and reproduction. Wow, very busy, okay. So, and then they reproduce by binary fission, okay, just separating the 2 parts, okay. Okay, they don't reproduce sexually, so their DNA is a constant, okay. And unicellular organisms do not have organs to breathe or undergo just ventilation. And no specialization of the cell, just one cell. Okay, right? Only unicellular cell with chloroplast undergoes photosynthesis. There's one cell organism that can produce photosynthesis. We'll learn one later. Right here. So it's example of a unicellular organism. This is 1, 2. First of all, for example, Paramecium gorilla, you need to understand where do they perform the 7 functions. You need to remember them. It will be asked in exam. Okay, these 2 are important, named in the syllabus, in the guide. Okay, Paramecium and gorilla are common unicellular organism, one cell only. Respiration and excretion for this one, okay, for Paramecium, okay, both absorb oxygen and excrete carbon dioxide by diffusion through the cell membrane. Very easy. For oxygen and carbon dioxide, both have a DNA process in the nucleus. They are eukaryotic. Cilia moves as a Paramecium. Okay, not shown here. There's a cilia here. Okay? Move it for movement. Okay, it also moves food and water into Paramecium through the oral vestibule. Vestibule get the nutrient. Okay, its function is movement and feeding for the cilia. It is too little. I don't mark it here. Contractile vacuole. Contractile vacuole. You can see some contractile vacuole, one at the front and one at the back. Okay, it is the maintain the water content. Okay, homeostatic. Maintain the water content of the Paramecium. Store excess water and expel them if needed. And then food vacuole. Okay, food vacuole digest and store food. Okay, digestion. Okay. And then endopause is here at the back. Okay, it ejects waste from Paramecium. Okay, its function is excretion. Okay, and it is an oral vestibule. We have discussed the cilia move the thing inside for entrance of food. Okay, it is for feeding. And then the gorilla doesn't need to be standing because it uses photosynthesis to produce glucose. Okay, but the glucose is stored as a starch granule, so it is does not feed or digest. Okay. Homeostatic, okay. Gorilla keeps its constant glucose content through the conversion between glucose and starch, okay. And gorilla does not have cilia or flagella. It does not move by itself, okay. It floats in the medium, water. And multicellular organism, a comparison between a unicellular and multicellular organisms are fairly easy. A multicellular organism has many cells, okay. So every cell can have its own function, right? Not all the function by it, so it has a specialization. Okay. Okay. So the cell can interact and forming different parts of the body inside a multicellular organism. Okay. But for the unicellular, just by itself, lonely. Okay. Do all the things. Okay. And but there's no general difference between the genes. Okay. For unicellular and multicellular, get the same for the gene. But the expression, the activity, the action of the genes are different. Okay. For example, a multicellular organism practices division of labor. Different cell has different specialized work. Okay. So different cell, although they have all the genes, okay, but the cell only part of the genes in the multicellular organism is expressed. We call it expressed, means it is active. Okay. So multicellular cell has all the genes, but only some of the genes is expressed. Different cell express different genes. Okay, but a unicellular organism do all the things, just one cell. Okay, so it expresses all its genes, okay, in one cell. That is the major difference. Okay, many cells work together. One cell, lonely, work that themselves. And number of nuclei. Okay, basic limitation of the cell theory. In cell theory, all cells comes from pre-existing cells, right? But the theory does not talk about what's the formation of the first cell. We are looking for it. Okay. And also, but the cell theory has some, but not necessarily all share characteristics. So every theory has limitation, right? So sometimes the theory has to eliminate viruses cell, okay? As viruses has some shared characteristics of cell also, but not all. Okay. So it is hard. There's a limitation for cell. For example, virus. For example, this one. Okay, number of nuclei is a problem for cell theory. Cell theory said that majorly one cell should have only one and only one nucleus, but there's an exception. For example, red blood cell. I draw it red, sorry. Okay, no nucleus. Okay, for red blood cell. Okay, and for muscle cell, multinuclear. And for SNAP mode, 2 nuclei also. big and small one. This one is important, we talk about it later. Okay? A red blood cell has no nucleus. A muscle cell has more than one nucleus. It's a very long one. Okay. A stem will also have two nucleus. Okay, micro and the macro. Okay, and a hyper is a tube-like structure of a fungi. Okay, no problem because normally the fungi has a lot of cells, and multiple cells partitioned by the cell membrane septa. So it's partitioned. So each cell has one nucleus, but an aseptor fungal hyper like this. The partition disappear. Low cell membrane, but still a multi-cell. Okay, but low cell membrane, so we have to take it as one cell, right? A long one cell, low partition. Okay, by membrane, so it becomes just a one cell, a long cell with a lot of nucleus. So we call it aseptor fungal hyper. It appears in the 204 May paper. So be careful. Okay, maybe see it in the new syllabus too. And then I talk about the cell theory. Why the cell keeps small? The concept is a surface area to volume ratio. What is this thing? It is a linear ratio between surface area over volume. For example, we use a cube as an example. A small cube of side A, bigger cube side 2A, even bigger cube side 3A. We observe that. Okay, what are the total surface area? 6A squared, right? Each surface A squared and the cube has a 6 surface. Volume A cube. So the surface area to volume ratio is that's the surface area 6A squared over A cube. Right? Cancel out the A, 6 over A. Oh, so when A increases, this ratio, the denominator increase, this ratio decreases. Opposite is negative correlated. Okay, so the size and the ratio is negative correlated. So what's the problem? Okay, think about the surface area and the volume. What are they to a cell? To a cell, the rates of exchange of heat, waste, and the nutrient happen at the surface of the cell, right? So it depends on the surface area of the cell, right? But what is their metabolism is inside the cell. It depends on the volume of the cell. So they do a lot of things if the volume increases, right? But they need a lot of more oxygen, carbon dioxide, or they need to excrete more waste. It depends on the surface area. So when a cell grows, getting bigger, the surface area to volume ratio decreases. It means that not that efficient to exchange material compared with the production and consumption inside the cell. So when it drops, the efficiency drops, there should be a limit for the surface area to volume ratio. So there's a limit to the size of the cell, okay? For example, the red blood cell do it by making it by concave shape, okay? Increasing its surface area and decreasing its volume. Okay? Increasing the ratio. Okay, this makes it transfer oxygen and carbon dioxide more effectively compared with the spherical size. Okay, and the huge size of cell. Oh no, sorry, cell theory has another problem again. Okay, another limitation of the cell theory. Giant algae. Okay, by cell theory, cells is the smallest unit of life, right? But unluckily, some unicellular cell can be very big. For example, for a giant algae, okay, one single cell can be a sphere cm and you can see it, okay, by naked eyes, okay. So it contradicts with the smallest assumption of cell theory, but it's just just one example. I think it's okay. And then we talk about for the viewing of the object using a microscope. Okay, higher magnification vs a higher resolution. There's these two concepts. Magnification means magnifying the thing, okay? Resolution means a clear difference, okay? For example, magnification of the microscope, everyone knows IP is the magnification times the objective magnification. It can be 1,000. So for example, we saw the thing here. For the normal visualization by the microscope, if we magnify the magnification, increase the magnification, we can see just this part. Make this part bigger, okay? And if we are buying a better microscope, the same magnification, we can increase the resolution. Using electron microscope, for example, seeing the inner structure. Remember, light microscope only see the compare different kind of cell, but we can't see the inner structure clearly. We need an electron microscope. So resolution is the clearest of the observed image. Of course, both can be greater is better, right? The greater resolution clearer, and the greater magnification, we can see a larger size of the observed image. Both are great, okay? And electron microscope do both, okay? Higher magnification and higher resolution. And recognition in the light and electron microscope are quite different. So remember, okay, in light and electron microscope we can see both distinguish both different kind of the cell. Okay, we can do so both, but only electron microscope we can distinguish the inner structure of the cell. Okay, so because of the advancement of the electron microscope, now we can see clearly about what's happening inside the cell. And then organelle in a micrograph. Sometimes you need to learn how to read the watch or read the micrograph of electron microscope. So after this page, okay, you can print it and then you go to internet, Google a lot of electron micrograph, okay, and try to recognize this one. Okay, there's a lot of membrane. Okay, sometimes it's thin, is the membrane. The thick line is the cell wall. Okay, and vertical is always a perfect circle because surface tension maybe of water. Okay, and mitochondria, remember, short lines. Okay? Colobus, long lines with dark segments. Okay, dark is the stroma, and nucleus is always round with a dark spot. Remember, nucleus, and the smooth and reticulum, smooth and endoplasmic reticulum is a parallel lines. Okay, and the rough one has some dots on it. Okay. For example, exocrine gland cells of a pancreas is a famous one, always happening in IBDP. Be careful, okay? It is drawing an annotated graph, okay, diagram. Remember, when you draw it, remember there's a requirement. You need to write down the function beside it, okay? For example, the thing is like that, okay? Wow, very big one. But this one is a famous, you need to remember it, okay? This one is a pancreas. Pancreas secretes a lot of enzymes, okay? And the enzymes are proteins, okay? So for this gland cell, okay, it has a lot of rough endoplasmic reticulum and Golgi apparatus, okay? And you saw the nucleus, rough endoplasmic reticulum, and after they made the enzyme, they are stored in the lumen. And after that, the lumen, they use the vesicle to transport out the enzyme. So you see the structure is like this, okay? It is not a micrograph. Micrograph is more a bird, actually. Okay, you need to use some wisdom, experience to handle them. So go to internet, look for them. Maybe you look at the 10 or 20 photos, and then you get some idea about it. Remember, the looks outside. Okay. Of course, another one is mesophile cells of the leaf. Okay, on the leaf there's a mesophile cell. You remember it because it's a big vacuole. Okay, you can see clearly. And then the other one is colobus similar shape. Sometimes you will see some a little bit dots that there's a mitochondria. And there should be a small circular, very circular nucleus you recognize. Okay. And then the other one is how to recognize the colobus. Okay, when they have a magnified colobus here, remember colobus has lines. Okay, and the lines is the dark part is the stroma. It's a size for photosynthesis. Okay, and then membrane here, and then you also see some breakable gray thing. Okay, they are the starch gland. They produce the glucose storage as a starch, and outside it, okay, you can see another membrane, and maybe some thicker one. Maybe thicker one is a cell wall. So it's a cytoplasm. So when you recognize it, it's like an arc. Okay. Observation. You need to learn to appreciate it. Oh, at this high level. Okay. So bye bye to the SL friend. Okay. So it is an endosymbiotic theory where how do prokaryote evolve to a eukaryote. The concept is that a big prokaryote engulfs some small prokaryote, and this prokaryote survives inside the prokaryote and becomes its organelle. Very funny theory. Excellent. So which kind of organelle have this property? That remember it has one membrane, cell membrane before as a prokaryote. So another membrane, so double membrane. Remember which one? Yes, mitochondria and colobus. So where is the before one? Okay, colobus, aerobic prokaryote, right? It's a aerobic respiration. Mitochondria is aerobic respiration, and the colobus is the one that they had the photosynthesis. So I mess is to exchange. Okay, sorry. So it says clearly, okay, evolve the prokaryote inside evolve into a eukaryote. We call it endosymbiosis. Okay, survive. Okay, so the large prokaryote engulfs a small prokaryote without digesting it. They survive and becomes organelle. Aerobic one becomes a mitochondria. Yes. Photosynthetic one becomes a colobus. Good. How do we know it? By the double membrane. Yes, double membrane. And later on, we have another theory also talk about how the liquid DNA becomes a nucleus too. Okay. A string of a theory. This theory is very strange. Okay, you can do a lot of explain all the observation, and then you can have some prediction. Okay, so because and endosymbiosis theory is quite good. Okay, so we trust that it is. We trust it's a good theory. Okay, we accept it. Okay. Now, even the exam question is about describing a theory. Be careful. You need to say some supporting evidence and implication of the theory also to get a good mark. Sometimes the questioning word is not that key. Okay, be careful for a theory, especially. Evidence supporting endosymbiotic theory. Remember, mitochondria and the colobus has a double membrane, and mitochondria and the colobus has the 70S ribosome inside itself, not the 80S. Okay, yeah, they're from another world. They still inherit the prokaryotic cell before the 70S ribosome. Okay, so we think that mitochondria and colobus are from other prokaryotic cell, and they do their own binary fission. Okay, so we trust that it is they are from other things. And they also mitochondria's inner membrane also manufacture ATP. Okay, similar you got prokaryotic bacteria membranes. Okay, and also mitochondria itself has a circular DNA, liquid circular DNA. So right, not that in the colobus. Okay, so we trust that they have just easily remember the prokaryotic stuff. Some of them are still remain in mitochondria and colobus, and we trust that it is the reflect that it comes from prokaryotic. And gene expression in cell differentiation, for HA, you need to learn more about it. For cell differentiation is the process for the development of the specialized tissue in a multicellular organism. How to do it is the expression become active of the particular genes. Okay, and they always triggered by the change of the environment. Okay, when the environment change, the cell will differentiate into different kind of cell. Okay, we call it gene expression. We'll talk about it clearly in later on about the genes. Okay, and different gene expression facilitate the deletion of labor of different cells. Okay, it helps, but not necessary because unicellular cell can survive by itself, right? Not necessary. Okay, one cell can do all the things actually, but now for multicellular organism, we can have more division of labor. Okay, specialized organs. Okay, and evolution of multicellularity. And when you talk about multicellular organism, we observe there's 25 major evolution. Nowadays we can have 25 at least. Okay, and this major evolution always come from the predator. Some predator appears and the sort of environment change. Okay, so the organism change. For example, some fungi and eukaryotic algae. Okay, and we saw that they change. Okay, some major changes. Okay, 25. Okay. Thank you. Thank you for going through it with me. So we learn a lot about cell structure today. Okay. So every part is important. It's the foundation. Although a lot of things we will learn about it more later. Homeostatic, gene expression. But it is a great chance for you to first glimpse of what is the requirement of IBDP biology. The story is much in details than the junior form discussion about when talk about in science or whatever. Okay, so we did it. Okay, see you. Bye.